OK, parfait.
Welcome to the Night Science Podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai
and I am Martin Lurcher.
So, welcome everyone. Today we're going to do something a little bit different.
Totally different from what we usually do, right? Usually we talk to great scientists about their process.
Right. And they tell us all their tricks and all their ideas about science. And it's really good, right? But then what did we notice, Martin?
Well, we noticed that certain topics popped up again and again. And there's one topic that we want to focus on today. Itai, you and I, we wrote an editorial with the title, "A Hypothesis is a Liability."
It's all about the dark side of hypotheses. They're great, but you could also miss the gorilla in the room.
Yeah, you could miss the gorilla in the data set.
Right.
And it's also about the relationship between data and hypotheses.
Right, because there's a special kind of feel that you have when you get data. You can find new ideas, you can explore, you see things you just didn't expect.
Yeah, and we realized that a lot of the stuff that our guests said was closely related to things we were thinking about in this editorial.
Right. And then in parallel, we got an email from a master's student whose name is Arjun Udupa. And Arjun said, "Hey, can I be an intern on the Night Science Podcast?
Can I help you guys?"
And we said, "Sure. Can you help us with making a special episode where we take all the pieces where different folks have touched on this particular issue, and we'll do a special episode on that?"
And Arjun listened to every single one of the old episodes.
Poor soul.
Yeah. And he picked out the bits that he thought were most related and most interesting about this. And he scripted a wonderful episode, with the things that Itai, that you say, and the things that I say, and then the snippets that we play in. And we tried to record that.
Arjun, we tried. We really tried, Arjun.
We did our best.
We did our best, Arjun. We're total failures because what ended up happening was we went off script, as it were, and just kind of, we're ad-libbing and improvising. And so what's resulted and what we'll start to play for you, dear audience, is really Arjun's script, but us riffing off of it.
So let's get started. Data and how we interact with data is really what distinguishes science from non-science. And different scientists have different ways of interacting with the data. And the first example we want to play for you is by Alan Rothenberg.
I think most of it is inhabiting the data. So if you are really trying to get a full understanding of a mechanism, then every piece of data that you get either fits or it doesn't fit. And that can be, in the old days, we used to do experiments that were really hypothesis-based and where every assessment of the samples that we made was based on a very focused hypothesis. Now we do a very large fraction of our work by sort of genomics methods, which give you this huge harvest of data, which wasn't explicitly part of your initial hypothesis. And It's there freely for you to play with. I think I drive the people in my lab crazy looking at genome browsers and looking at gene lists because I'm always trying to cluster them and put them together into pathways that I think I already know about. And I start jumping up and down all the time when I can find something that looks as though it's showing us either a new conjunction or a failure of a conjunction that we thought was there. And it's always trying to live inside the data and use it to test the structure.
Yeah, inhabiting the data, I think, is such a good way to put it. When we talked with Manu Prakash, he talked about immersing himself in the work. And that really resonates with the way I see things, that you have to be in a state where you're inhabiting the data, you're immersed in the data. And when you do that, you can really transcend any particular hypothesis that we impose on it, but just be open and let the data do the talking and tell us what's new.
How do you think this concept of inhabiting the data is related to the idea of becoming your object of study, like being the transcription factor?
Being the transcription factor, immersing yourself in the topic. It's the same thing. The whole notion is that you're exploring The whole notion is that when you have a hypothesis, in a sense, you're looking from the outside in. You're imposing a particular idea. But if you're able to let that go, then you can go from the outside and step into it. And now you're just in the topic, you're in the data set. And that's a great way to let go of any kind of biases that you may have.
And this power of, just inhabiting the data, being open, is something that's shared across many different fields and subfields, right? For example, the story that you and Bernie told us.
Sometimes the things that you think are artifacts are, of course, really interesting bits of biology. I had a great example of that. I would not bore you with the technical stuff, but there's a very common thing in genomics called blacklisting. So these are parts of the genome which are behaving weirdly, and blacklistings are parts of the genome which you just sort of blank out for this assay. And so my student came to me and we were looking at this. The null model refused to fit, and there were like 1,000 points that were just like stubbornly not agreeing with the rest of the data points, which were all looking beautiful. And, we were going back and forth with different blacklisting. And I eventually said, look, if you can't work out what these things are in a week's time, we are just going to create a very specific blacklist for this assay, and we're going to blacklist all these guys. Okay, so that's it. Kind of decided that these things were artifacts. I just didn't understand them.
Okay.
And at the end of that week, he came back to me and he said, well, yeah, okay, I'm up for this, but I just want to check you. They're all on the X chromosome. I'm like, really? They're all on the X chromosome? And I was like, okay, let's look at that. And they're all on the X chromosome. And then if we split them by male and female, our model fits. I was like, okay.
That's something.
That's something. So then we have to completely turn the car around, dig into this thing that we thought was an artifact.
I love that. They made a discovery without having any hypothesis in mind. And just like Alan Rothenberg, they had an outlier, something that just didn't fit with the rest of the data. And instead of doing what most people would be at least tempted to do, just ignoring this boring outlier, right, that just ruins everything. Instead of doing that, they just dug deeper into it and explored it.
Yeah, I think all good discoveries start out by saying, what's this? This doesn't make any sense.
Exactly. There's something wrong here, right?
Either there is something wrong, it's just an artifact, and then you need to address that too. Why is there an artifact?
Of course.
Or it's actually something real. It's an outlier, and you need to embrace the outlier. That's how George Church teaches the members of his lab. Openness is your friend. I like that. I also like your meme, we can call it, of embracing the outliers. It reminds me, Daniel Segre, your former postdoc, now a professor at BU, he's an old friend of mine, and he once told me that the number one thing that he learned from you is to embrace the outliers.
Oh, yeah. I taught and teach occasionally computational biology courses and, draw a plot, scatter plot. Well, there'll be one point that's way the heck off of the scatter plot.
Yeah, what's that?
Well, you could come up with a filter. The filters are a really popular computational biology thing, or
right,
statistical tool. Or you could drill down on that point and figure out what's different.
Right.
It's either going to tell you something about your experimental methods that you should fix so you don't get artifacts, or is going to tell you there's something novel here in physics, chemistry, or biology, or both, that maybe before you fix it, you figure out what it is. And, it could be anything. There's often outliers, and we often ignore them. When I was doing crystallography, we had a case where there was a bond missing in the transfer RNA. There's no bond there. And it turned out that the heavy atom that we use for solving the structure actually caused the RNA to cleave itself at that place. And so we had basically discovered RNA enzymes, ribozymes. We didn't publish on it. We didn't even think it was significant. It was just a broken bond that showed up in our electron density. And then a similar thing happened to Tom Check, is he's trying to make some RNA and it keeps getting spliced. It gets contaminated with some splice form. And both of those were outliers, and he paid attention to it. That was the difference.
Yeah, if you ignore the outlier, you might cheat yourself out of a discovery.
Exactly.
The thing is, though, Martin, when we talk about exploratory analysis, we have to remember that not everybody has a high opinion of it. like we do. Other people derogatorily call it a fishing expedition. When someone says, oh, you're off on a fishing expedition, they don't mean, oh, you're doing great science. They're criticizing you for not having any direction. They see you as being aimless. And yet, if you ask, where do the most groundbreaking discoveries happen? It tends to start with exactly that kind of exploration.
But you know what? If you would do a stupid fishing expedition, right, you would go out on a boat and throw your net out in a random place, probably you're not going to catch very much. But if you're a good fisherman, you know where to go. You'd go to the right place and you throw your net there and you're going to find something, some interesting fish. So it's really about where are you going to fish?
The art of fishing expeditions. It's true. You have to have some intuition about where to cast your net.
Yes.
Then an initial hypothesis-free exploration is a really great way to get new ideas, to get new hypotheses. And so that's why they say that fishing expeditions are a great way to catch fish.
Absolutely, absolutely.
It's a way that's not only powerful, but it's actually pretty beautiful.
Yeah, I really liked your meme about that you recently published on X. My hypothesis is that if I put together this data set, I'm going to find something amazing. I like that.
Like in the old days, that's what we would say. We would just go into data sets and just try to find something cool.
Let's listen to a conversation about fishing expeditions with Tsachi Pilpul.
I actually don't recall being accused of doing fishing expeditions, and it's not because we don't do them. I was prompted by you to think about that in retrospect to see, what would I consider a fishing expedition among the works that we have done in the past, maybe the ones that I appreciate the most. So I'll give you examples of something that started as a fishing expedition and turned to be something very profound. Several years ago, Anders Lund from Copenhagen was here at the Weizmann, and he showed us his microarray for non-coding RNAs in the human genome. And incidentally, he had a few probes for tRNA genes that he hybridized to hundreds of cancer samples from patients. And I asked him, can I just look at the data because I care about tRNAs? And he said, yeah, sure, take a look, because we couldn't make sense of what we see with the tRNA. Maybe anti-sense, but not sense. And we started to look at the data, and maybe one of the most important discoveries for my lab came out through a collaboration with Lund. It culminated in a Cell paper in 2014 that we discovered duality of coding of human genes and how tRNAs take advantage of this duality in cancer.
Analyzing data, even data that's previously published, relating it to other data sets, it's just a great way to do science. And a lot of people these days are saying you don't even need to generate your own data set because so much data is already out there. There's so much to explore. And even someone with very limited resources can make tremendous discoveries by just thinking creatively about data sets.
Absolutely, absolutely. I mean, the journals sometimes don't appreciate that as much if you don't bring data.
Yeah, the iron rule.
Yeah, of course. The iron rule of science. The only way to judge if something is true, the only way to convince your colleagues of something is by bringing data. But anyway, another take on fishing expeditions came from Per Borg.
Quite a bit of a group is involved in large collaborations. And there you have more defined tasks. It's more data-driven stuff. And then we have our own potpourri of projects. The idea is to be very broad and not so deep. But then if you find a nugget, you zoom in and you add biology to the detail. So we often work in emergent fields where you have uncharted territory. And then you have to get a feeling what's important, what's not important. And one way to make such a decision is really thinking of, could I tell the story to a non-scientist? Would she understand what we are up to? So that's a bit of pre-selection of things if you want to go for important discoveries that the public understands. And usually the important ones the public does understand. And this is a way how things also unfold. You have a set of data and, roughly the biology around it. And what are the questions that nobody has thought about? That's the ambition.
That's really interesting. You said you could have these broad searches and then hope that you would find a nugget. Do you have a trick for knowing when it is that you find a nugget? Because, one person's nugget of gold could be another person's garbage.
I have a biological background where I can see there could be interesting biology. Either it is against a dogma, which is not really proven, but the field believes it, or it's something very simple.
We want to also really show our appreciation for our sponsor, Research Theory, who has supported in part the making of this episode. Research Theory is this nonprofit that has a goal of improving creativity and the culture of science. And of course, that's something that we feel very passionate about ourselves. So our interests are really aligned here. And we're so grateful to Research Theory for supporting us.
Yeah, and check them out at researchtheory.org.
You know what's interesting to think about, just as an aside, is right now, I totally agree with you. Journals will respect a paper more, be more likely to publish it if they come with data, right? Now, let's imagine a ridiculous scenario where all the possible data has been generated.
Yes.
Okay, so there's no data to generate because we built this country with all robots, and the robots, they created every possible data set. Okay, so like whatever you imagine, you just log in and you get that data set. Okay, now who gets the good publications? Now, well, then it has to be the people with the best ideas, right?
Absolutely. Yes.
And so wouldn't you say that the logical conclusion from that is that already we should be giving our greatest attention not to the papers with like the most data, but to the papers with the best ideas?
Are you trying to convince me? I mean, of course it should be about ideas and not about who generates the biggest data set and the most expensive data set.
Yeah, there's been a kind of commodification of science. Okay, that was just an aside. So your conversation with you and Bernie, that's on a similar theme.
My colleagues and myself sometimes get hit over the head with this in peer review of our grants. Fishing expeditions is the claim for genomicists who are just generating data. There's some validity to the critique, which I can come to, but mainly it's not a valid thing. I think the best way in this discussion of hypotheses is a very classical way of thinking about science and being able to refute things, is to say that you're in the hypothesis generating part of this process.
Right.
So what you're doing when you gather data, you basically say, I think if I gather this data, I will discover things, and here are some ways I'm going to analyze it, which I have some confidence that it's going to work out. But ultimately, I will generate hypotheses, and then I can test those hypotheses. Now, when I think it goes wrong is when the fundamentals of how you gather the data cannot possibly answer some of the questions that you want to answer, or cannot generate valid hypotheses. So, we can measure all the genes expression in a cell type at once, or all the protein phosphorylation sites at once, or we can survey all the genetic information in the genome in a single shot. And this sort of comprehensive view of a particular class of phenomena does give biology a bit of a joker in the pack of asking questions, because you can really say, look, it's got to be somewhere here. I've set things up correctly. It must be somewhere in my data set. So you can use that comprehensiveness in quite clever ways.
Yeah.
So to that kind of, do you need a hypothesis? I mean, I've literally had to do this a number of times where I've had to state your hypothesis, and I've had to state, humans are a complicated species. And, my hypothesis that by generating this data set, correlation patterns of this type will emerge, and they will correlate to things which are about the fundamental molecular biology of the thing you're looking at.
I really like that formulation. I don't think I would have come up with the idea of calling that my hypothesis.
Yeah, I mean, I think although this gets into an epistemological philosophical discussion about what do we mean by knowledge and understanding and la 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 la, but in practice, I certainly move between discovery and hypothesis generation and hypothesis testing and triangulation. So this is a really good concept, triangulation. You've made a set of discoveries, or you've generated some hypotheses about how the world works from this big data set. Now, can I bring an orthogonal data set, which is clearly not related to the data set that I used to generate this, to test that? And obviously, sometimes you might do a very deliberate experiment, and that's the most obvious way to do that. But in fact, you don't have to. Sometimes you can bring a data set which you can convince yourself and your colleagues is completely separated from your first data set. Right. And yet it interlocks with your data set in a particular way.
And it can kill the hypothesis if it's wrong.
Yeah,
it has the power to do that.
What Ewan means with triangulation, you've used the data set to find something, to discover something. So now, of course, you can't use it anymore for testing the same thing. That would be circular. So you need an additional data set that is independent of the data that you've looked at so far.
And this gets us out of the problem that most people cite about exploratory data analysis. They say you can't do two things simultaneously in a data set, both find a new idea and test it. Because if you do that, if you don't have a pre-registered hypothesis, if you are just looking for patterns, you could be fooling yourself. You could be finding something that's really only true randomly in that one data set. And so they say don't explore. However, then you don't have new ideas. So I think triangulation is the perfect way out of it because it says, okay, explore the data set, look for new ideas. It's going to be okay. You're then going to test it on another data set.
So the exploratory data science, that gives you the idea. That's the night science part. And then the triangulation with the independent data set, that's the day science part. That's the adult in the room that checks if what you thought you saw in the exploration is actually
real. Exactly. And that's what a lot of people don't realize when we say night science. We don't mean, heavens forbid, that you get rid of the robustness. No, it's just that we make room for another mindset which gives you the ideas in the first place.
I mean, of course, you have to have the idea of which data set you're going to use for the triangulation, and there's night science involved in that. But then the essence of triangulation is that you want to test your hypothesis. I mean, that's the core of day science. And that's what makes science so powerful. And I just want to reemphasize what you just said. It's not that we think day science is unimportant or boring. It's just that it's not enough. You need to have hypotheses before you can test them. But of course, explorations aren't limited to just huge data sets. You can do exploratory data analysis even at much smaller scales, as told to us by Edith Hurd, the director of the CRIC.
For those 10 years where I was testing what is the region of the X chromosome that triggers X inactivation. And there were papers that came out that said, this is the minimal region. And I would do my tests using big transgenes. And I was finding that actually single copy transgenes didn't work. So I had to come up with a whole bunch of hypotheses, like, could it be that, a single copy transgene doesn't get positioned in the right place in the nucleus? Could it be that the single copy transgene is not creating the right chromatin state? Could it be that the single copy transgene is not being seen by the other copy of the X in the same way? So I had this whole bunch of hypotheses and no way to test them. And I actually went then to Cold Spring Harbor and to David Spector's lab in particular to test some specific hypotheses that I knew I didn't have the technologies for. So that's what I did. And then within a month of getting there, Dave Alice, who very sadly passed away and who was an amazing person and inspiration for me, he wandered into David Spector's lab with a bunch of antibodies in his pocket. It was the days of the histone modification antibodies that, Dave Alice and others were just starting to produce. He plopped them on the desk and said to Dave Spector, one day you should test these out. They're really cool. So David Spector, just put them in the fridge. And I went up to David Spector. I said, could I test them? And he said, sure. And so I did the experiment. Dave Alice was still at Cold Spring Harbor. He'd come to give a seminar. And that night, we all went out to dinner, actually, with Dave Alice, David Spector. And we were laughing about, who knows, maybe she'll see something. The next day, I looked on the microscope, and it was incredible. I had never imagined that there would be an enrichment of one mark and a depletion of the other mark on the inactive X. I hadn't gone to David Spector's lab to do that. It was complete chance that Dave Alice happened to come by. And that was the beginning of a whole new series of experiments. And I actually ended up getting very involved in the early epigenetic changes of X inactivation, which was not at all what I had intended to explore when I went to that lab. But nevertheless, that discovery of these marks led me to the understanding and the realization that some of these chromatin changes might actually be the explanation for why my famous single copy transgenes were not working. So then I actually started to go back and test those hypotheses. And it turned out that indeed, there probably was a whole region that was enriched in heterochromatic marks that was probably involved in helping the exon activation centers, the key locus, the key trigger, to do what it did. And so in a way, you need both. You need the hypotheses that you go and test, but then you need to have the open mind to pick up the unexpected opportunities or the unexpected observations and follow them up without fear. And I honestly think that if I hadn't been at Cold Spring Harbor, I never would have ended up having the career I had. It was that it's okay to try this, and it's okay. Whatever you see will be potentially interesting, and it's okay to follow it up and to think out of the box. So for me, that was a real eye-opener. That year in my career was truly transformative.
Yeah, so isn't it amazing? She really used these histone markers, this new tool that fell into her lap, for absolutely no good reason when it comes to day science. She had no hypothesis that she was testing. So what was she even doing? But she just did it. And it opened up to her a whole new world for hypothesis testing, new ideas to now consider.
Yeah, because she had exactly that night science attitude of just being open, of just saying, yeah, let's try it. Why not?
Let's just try it.
Yeah, there could be something interesting there. Let's just see. At the time, it made very little sense. And it's easy to imagine that any rational person would talk themselves out of doing this, right? Because it could be seen as a waste of time. But this openness, this preparedness for trying something, even if it's not related to any hypothesis you have, and even if you don't know if anything useful is going to come out of it, that's the basis of many discoveries.
I like that notion of imagining a rational person talking themselves out of doing night science, because the night science does require this leap into the unknown, right?
Into the irrational.
Into the irrational. Edith Hurd, when she talked to us, she actually highlighted that the Americans do this very well, if I may, which is that they have this attitude of just try it. Just try it. Who knows? You may not need a very good reason, but just try it. See what happens.
And, explorations are one of the great night science tools. Just looking for patterns, looking for connections. It's extremely powerful.
Yeah, because finding patterns helps us to generate new science ideas. And when we talked to Rich White, he told us how he clearly benefited from seeing patterns in what a normal zebrafish embryo looks like, which then allowed him to identify abnormal phenotypes.
Yeah, I guess it kind of relates to things you've both talked about, is this question of hypothesis. Not that I think hypotheses are good or bad, actually, but the random walk and generating data to just stare at is really informative. And for me, I remember moments that I've had this, look at a dish of embryos. They all kind of look the same and they're all kind of marching along doing their thing. But then you have one embryo that looks a little different, like a little cell's crooked or it looks weird and stuff like that. And you're like, I wonder why that is. And the only way you learn that is by just staring at a lot of data. Just stare at a crap load of embryos. And you'll kind of begin to realize, 99% of them are just going to march along and do their thing, but 1% are going to do something a little different. What is it about those 1%? And so, I think I do kind of search for it in a lot of ways. But the only way to search for it is to be a bit hypothesis free and just stare, just look and just stare for a while.
Like Ellen Rothenberg said in her podcast episode, you really need to have a very good detailed mental representation of the system that you're looking at, of the knowledge about that system, in order to discover something as being unexpected.
There is a requirement to be very ambitious in one's thinking in order to see where the current structure of knowledge doesn't fit. And I think in this sense, I kind of agree with T.S. Kuhn in The Structure of Scientific Revolutions, that a lot of times you do reconfigure the way you understand something when you realize that the prevailing structure of explanation doesn't fit. And the interesting thing about that is that what really helps is to be very explicit, very clear, and very detailed about what the prevailing structure does fit. And that way, when something really doesn't fit, you can see that fact. I think one of the things that I worry about with some younger people who've come into our field at a time when it was so data rich, they actually have a kind of a passive defeatism about that. They don't necessarily feel that they ought to be able to understand everything. And therefore, when something comes in that actually could be the key to a major reconfiguration, they don't see it because they don't have a well enough articulated sense of what it is that it would violate.
But of course, on the flip side, if you're looking for patterns, humans can't help but find patterns everywhere, even when there are actually no patterns.
Like in the clouds, we look up at the clouds and we see our pet.
Or whatever we see there. People call that patternicity or apophenia. And of course, that makes us vulnerable to being fooled by randomness when we mistakenly infer relationships between things that are generally independent. And it's obvious that spurious results will be generated when we do unguided explorations.
It's true. It's really important to understand that day science has a liability of missing new ideas, but night science has its own liability of being, as you put it, fooled by randomness. You can see all kinds of patterns that simply don't reflect anything true. And so what we need to do is constantly switch back and forth between night science exploration, finding these correlations, and day science testing, being the adult in the room and robustly asking, is this true? This is the eternal conversation that we have in science between the data and hypothesis. Data, hypothesis. When we talked with Sarah Teichmann, she spoke about this.
Well, I think there is a perceived tension, that there's somehow a difference between the manner and the value of the conventional hypothesis-driven sort of mechanistic studies as one molecule at a time, and what does it do, and dissecting each element of a signal transduction cascade or something like that, and the large-scale, high-throughput genomics research, that we are in, or computational research. But personally, I think that the distinction is actually misleading, because I'd be interested to know also what you guys both think. But to me, actually, both types of science include both hypotheses and data generation and interpretation. And you're not just generating genomics data in a vacuum. When we decided to study the placenta and the decidua, we had a very specific reason that we were taking that tissue. And the reason was that we wanted to understand maternal tolerance of paternal antigens. The way you construct the models, the way you apply them, you have some kind of mental model of the data and the questions that you're asking. So to me, there's a constant interplay between, hypotheses, data generation, data interpretation. It just goes round and round.
Yeah, she says it really well, that you need both. You need data generation and you need data interpretation. It's also just like what you and Bernie said earlier, that when you generate a data set, you have this idea that there's going to be something interesting, and that it's not just random data that you're generating. You have this strong hunch of where it's worth looking, that you have this intuition, and this intuition is something that you fine-tune over time. But then you also have to be open-minded enough to figure out if there's something new that's happening, keeping an eye out for the unexpected.
Yeah, to find something really cool, it's good to know where it's worth looking. So you need a design to get started. But then when you analyze the data, then you need to be open and in some way just let your mind wander so that it might find something that you're totally not looking for, that you're not looking in a certain direction. And I think it's this interplay between the two types of minds, the day science exactness and precision and the night science openness, that drives the whole science thing forward.
Martin, not everybody agrees with us on this perspective. When we talked with Bill Martin, he offered a cautionary view about this kind of exploration.
If you follow the data, then you've completely lost control. All right. Then you're putting the data where your mind should be. Okay. So
what do you mean?
What do I mean? If you follow the data, then you're letting the data dictate your thoughts. For me, the scientific process is one of questions. And these questions come from biological phenomena, okay? Biological observations, things that we can observe in nature. But if you go into data-driven science, like, oh, what can we find in the data? Seek and ye shall find. You'll find a lot if you go down that route, right? It's a lot better if you kind of know where you want to be at the end of the day.
How do you know that?
I don't address the data unless I have a specific question.
So what is Bill saying?
I think its last sentence says it all. He doesn't address the data unless he has a specific question, right? That's one way of doing science. It's a great way of doing science, but it's definitely not the only way. And if I may say so, Itai, it's not your favorite way.
I think Bill and I are thinking in different ways. I think for Bill, he also does the same kind of explorations that we're talking about. In my mind, though, he just does it where the data set is different. For me, for example, the data tends to be like gene expression data set. That's what I like to explore, right? I think for him, it's the literature. He goes to the old papers, and that for him is a way of exploring. And then he gets the idea, he wants to test it. He collects a data set expressly to test that idea. And then maybe he'll go back to his literature and get more ideas. So I don't think there's like a real contradiction. It's just that what we call data for him is just for the triangulation testing.
Yeah, but I actually think that some of the great work he does is comparative genomics. He tries to understand what happened at the origin of life or the origin of eukaryotes. What was the metabolism like? Like, what did these creatures eat at the origin of life? And to answer these questions, he compares genomes and he tries to figure out which genes did these creatures have and what did they probably do with those genes.
I don't know, that sounds like exploration to me though, no?
Yeah, he went in with a very clear question, but I wouldn't call that a hypothesis. It's a question.
Yeah, so I'm a bit confused then why Bill would say that he doesn't explore data.
I think what he says is not he doesn't explore data. What he's saying is he doesn't explore data aimlessly. He doesn't say, I don't address the data unless I have a hypothesis. He says,
Right.
I don't do it unless I have a specific question.
Oh, well then that's something I could very much relate to. And what we've always said is that you need to know a lot in order to explore. It's not the low road of science where you don't know what you're doing. You actually need to have a well-formed picture of the field. So I can understand what Bill is saying in that respect.
If you already know a lot, you know exactly how to pose your questions. And Bill knows a lot, and he knows how to ask good questions. So, to wrap up, there really needs to be a balance between hypotheses and data and exploratory research.
For me, the bottom line has always been that day science thinking and night science thinking are both required because they complement each other. As we've said, day science has a liability. Hypotheses are a liability because you could miss the gorilla hiding in your data set. Now, of course, night science has its own liability of patternicity. You could see things that are simply not true. And so it's this complementarity between the two that allows good science to happen. New ideas and then testing of them, back and forth, the eternal conversation.
Yeah. And as our guests have made very clear, I think, is in night science, when you're exploring the data to figure out what could be your hypothesis, there's no rules. Do it whichever way you want. If you come up with something interesting that you can then triangulate, awesome.
Overall, I would say explorations, night science, it's an attitude. You're allowing yourself to be open to something you did not pre-register, you did not conceive of, you didn't hypothesize, you didn't do anything fancy yet. You're allowing yourself to just be open to something. Would it kill you to be open, Martin?
No, would it kill you to be open? No, and remember what Ewan said about this. You're in the hypothesis generation phase. It's okay.
It's okay. Keep calm and explore the data.
There's a few things that we want to tell you. The first thing is, it would be great if you would follow us on X, Blue Sky, and Instagram, where you just need to search for Night Science Podcast.
Night Science is produced by our great producer, Lou Barron, and also our audio engineer, Christoph Schneider. Martin, we've come a long way from the time where it was just the two of us working in our garage. We have a whole team.
Including a graphic designer who does also video editing, Kendall Martin. We have a great team by now.
Right. What could possibly go wrong?
Yeah, what could possibly go wrong?
And if you like this special episode, let us know, and we're going to produce more of those.